So hi there. Now let's talk about inhibiting the spread of cancer in the human body or repressing the proteins that we have structurally modified. So first of all, what we need is we need a protein structure. So we already got that. After getting the protein structure, we need a inhibitor. So if you are new to bioinformatics or as the industry, you should know that EF, EGFR are the protein inhibitors that are used to inhibit the growth of lung cancer. So I'm just gonna search it on the PubChem repository. So what is PubChem? PubChem is a repository that includes the ligands, which are inhibitor, enhancers, and things like that. So we have the compound EFGR inhibitor. So I am just gonna click on search on the PubChem, and after that, let's just see EGFR, and then click on this tab, and here we will see the results of this. Here we have the 2D structure of the ligand, 3D structure of the ligand, and we can use this structure to see the impact which it made in our research paper in our protein structures. So we will see where the results will be generated and things like that. So now that we are talking about the inhibitor, ligand, and protein, we should know where to dock these proteins and these structures. So first we are going to look at a database which is SwissDock. So SwissDock is used to dock the ligand and protein interaction. So what we need to do, we need to open the database. You can search on the internet and then open the SwissDock protein and then click on submit docking. After that you have to search for targets. So you can upload the file. So here you can choose file and then upload your model which we determined in the previous results. After that we have to select the ligand. For example, we can search for ligand, and here it will shows us the ligand is previously identified and it is present in the PubChem history. So we will select and dock one selected ligand, and then it will carefully perform its inspection, and then the successful setup will be done. So you can also inspect it as well. After inspecting, you can see all the things. After that you will enter your job name and then enter the email address. After that you can select the docking type. Here you can select very fast, or accurate. So accurate is good if you want to get the better results. So after getting the better results, you will click on the start docking section button. After that your result, you will see a window where they will be saying that your docking has been successfully submitted, and you can download the result from this tab. So I am gonna show you, but it takes some time to dock online. But if you want to use some other softwares like MOE, Pyrex, you can use that as well. You can see the tutorial all over the internet to how to use the docking of that compound. So after docking that compound structure, we can see that we get the results of these protein docked with specific ligand. So I have submitted earlier this and downloaded the docked protein with the structure EFGR structure as well. So here we can see we have our protein here, and a structure is allegedly docked here. So how we can determine that this is docked to the protein and it is not in the 3D space. So here you can see if I select atom and bond, so they are attached to bonds. So we can see that this is providently attached to the binding sites. So we will open our structure again and then make it more appealing and make it more interactive. So here we can see if we select preset and then select this rounded ribbon and select the okay, if we select the rounded ribbons, and also we can inspect the interaction as well by focusing on that. So if we move to tools, we can also see where the uh, atom is docked. So we can also align the proteins with other proteins by using this. So after doing this, we need to find the proper way to visualize this to include it in our research paper. So for that, I'm gonna show you some solid mesh of the protein to see where our protein is docked. So if we click on our surface and then click on show, so here we can see the protein is docked here. So what we are going to show the mesh of the protein. So it is visually appealing, the protein, and the interaction is similarly showing somewhere like that. So I'm just gonna keep it like that and click on focus. After clicking on focus, we can see that it is rather good to be a good showing of this. So we can also use this for the general picture saving. So if we move to the side view of the protein, we can see that the atoms are very good visualizing. So if we move to the lightning and the shadows of the protein as well, so you can see that. So we will make some changes to the figure where it is somewhere good. So effects, you can also add effect if you want to, but if you don't want to, you can just disappear it like this. So we will just keep focus on this, and you can use the camera angle as well to view at a better angle and then focus it to the protein section. So here is the whole figure of our protein where the element is docked, where our inhibitor is docked. After that, we will save it to image docked .png. So it will be saved to our desktop. So what is the basic thing that we need? Why we need to dock the protein? So it is totally upon you. So the reason why I chose the upregulatory gene and docked with an inhibitor is to see that if the ligand is correctly docked and it down regulates the promotion of those genes. So you can validate these results by using the simulation. So simulation can be done by using by using the tools installed onto your Linux operating system. So you can validate through there, or you can done it by hiring some companies to do that for you. So that's pretty much how you validate your structures and how they were causing the proteins structure alignment and protein structure significant for you. So here we got the results of our protein structure prediction and protein structure docking. So I'm just gonna leave it like that, and here we can see we have the results of our analysis done. So that's how you can use the protein structure docking, and in the next video I'm gonna show you how to change these results into proper research paper. So I'm just gonna highlight some things that you need to do for the proper write-up of this paper. So thank you so much and stay tuned for that.